Hello, and a very warm welcome to Cognito Key. Inside every single one of our cells, there's a microscopic superstar, a master builder that tirelessly constructs the very blueprint of our existence. Today, we're diving deep into the world of this incredible enzyme, DNA polymerase.
So, let's set the stage, shall we? Every time a cell in your body decides to divide, it's faced with a truly mind-boggling task. It has to make a perfect, flawless copy of its entire genetic instruction manual. And trust me, we're not talking about just a few pages here. This is a mission on an absolutely monumental scale.
So, here's the big question, right? How on earth does a cell pull this off? How can it possibly copy a library with 3 billion letters with almost perfect accuracy, again and again and again? Well, the answer lies with our tiny molecular hero.
Just think about that number for a second. 3 billion. That's the mission, to copy 3 billion base pairs of DNA without making any critical mistakes. To do that, you need unbelievable speed, you need absolute precision, and you need a built-in system for fixing errors as you go.
All right, so who could possibly be up to a challenge like that? Well, it's time to properly meet the master architect, the chief builder inside our cells that makes life as we know it possible.
Our hero is an enzyme called DNA polymerase. Its main job is to read an existing strand of DNA and then build a brand new, matching one right alongside it. It's like a tiny, super efficient machine that just zips along the DNA, adding the right building blocks, we call them nucleotides, one by one to create a perfect copy.
Now, this is where it gets really cool. DNA polymerase basically has two superpowers that seem like they're complete opposites. Its main power, the polymerase activity, is all about building, but just as important is its other power, the exonuclease activity, which is all about proofreading. It can actually go backward, snip out a mistake if it adds the wrong piece, and then replace it with the right one. It's a builder and a quality inspector all in one.
But hold on, this master builder isn't a solo act. In simpler organisms like bacteria, which we call prokaryotes, there isn't just one type of DNA polymerase. Nope, there's a whole team of specialists, and each one has a very specific job to do. Let's meet the crew. First up, we have the superstar of the team, DNA polymerase III. This is the main engine, the workhorse of DNA replication. It's a really complex machine made of 10 different parts that clamps onto the DNA and just flies along at an incredible speed. This is the one doing all the heavy lifting. Next in line is DNA polymerase I, or pol-I for short. Think of this one as the meticulous cleanup crew. After pol-III has done its high-speed job, pol-I comes along to fill in any gaps, repair any damage, and this is super important. It removes these little temporary starter blocks called RNA primers, which are needed to get the whole process going.
And rounding out our main team is DNA polymerase II. You can think of pol-II as the reliable backup system. Its main job is DNA repair. So if the main enzymes, pol-I or pol-III, get into any trouble, pol-II can step in and help out, making sure the genetic code stays intact.
So we've met the high-speed builder, the repair expert, and the trusty backup. Now let's put them all side by side to really see how their special skills stack up.
This table really lays it all out, doesn't it? What's fascinating is that all three of them can build and all three can proofread backwards. But look, only pol-I has that unique ability to also remove things in the forward direction, which is how it gets rid of those primers. You can see so clearly here that pol-III is the star player in replication, while the others are the repair specialists.
Okay, but this incredible story doesn't just stay inside a tiny cell. Oh no, our understanding of these little enzymes has led to some truly revolutionary technologies that have completely changed our world. Let's see how this tiny builder made the giant leap from biology to the lab. You see, for scientists to use these enzymes in a lab, they're looking for a few key qualities. Fidelity, that's the accuracy. Processivity means it can keep going and going without falling off the DNA. But it was a third trait, thermostability. The ability to work in extreme heat that really opened the door to one of the greatest inventions in modern biology.
The big breakthrough came when scientists found a polymerase in a bacteria that lives in boiling hot springs called Thermus aquaticus. Its enzyme, which we call Taq polymerase, doesn't break down in high heat like most others do. And believe me, this one single property changed, well, everything. This led to the invention of PCR, the polymerase chain reaction. It's a brilliant process. In the lab, you use high heat to split the DNA strands apart. Then you cool it down a bit so primers can attach. And finally, the heat-stable Taq polymerase gets to work building new DNA. By just repeating this heat-cool cycle over and over, you can make millions, even billions of copies of a tiny piece of DNA in just a couple of hours. It's just amazing, isn't it? From diagnosing diseases, to forensics, to genetic research, the things we can do with PCR are incredible. So as we finish up, just think about this. These tiny molecular machines build and repair all life as we know it. It just leaves you wondering, what will they help us discover and build next?
I really hope you enjoyed this deep dive into the amazing world of DNA polymerase. If you found it interesting, please subscribe to Cognito Key for more explainers like this one. And of course, don't forget to like this video and share it with someone who might find it cool too. Thanks so much for watching.